Okay. Good morning or good afternoon, everybody. First of all, I would like to thank the organizers. I'm very humbled to be invited to give a talk today. And I'd also like to thank all of you for staying this late or starting early to join this talk. I'm thrilled for the opportunity to share some of our work with all of you today. So I will talk about some of the computational tools we have developed within Agile Biofoundry and Joy Bioenergy Institute to address the needs and characteristics of synthetic biology with the goal of speeding it up. In particular, I will talk about two machine learning-based tools and show some use cases and how they integrate with other tools that we have been developing at IBF and JBay. So synthetic biology expectations are high. Nevertheless, synthetic biology has the potential for transformative impact in society. Here are some of the more recent examples of applications. Synbio produces food. It has been able to create burgers that taste like meat but are entirely made of plants, plant-based dairy, zero-calorie sweeteners, honey without bees. Synbio produces fabrics such as spider silk, synthetic leather with mycelium, or bio-nylon. Synbio produces health products, human collagen or anti-malarial drug artemisinin, diagnostics and therapeutics, or even combining the two, fabrics and diagnostics, to create wearable diagnostics. And finally, biofuels, such as farnesene, which is a diesel replacement that is used to propel the bus fleet in Rio de Janeiro. However, bioengineering is not easy. We gather data faster than ever, for example, through mass spectrometry, collecting proteomics, metabolomics, through microfluidics, cell manipulation through light, genome editing platforms, robot liquid handlers, but we need better predictive power. For bioengineering to become a major engineering discipline, there is a need for better predictive power. And our approach is to tackle this problem by using machine learning. And for that purpose, we developed several tools. As pointed out by the State of AI report for 2020, publications involving artificial intelligence methods in biology are growing. There were over 21,000 papers in 2020 alone. They include different AI techniques, leveraging data from cellular microscopy, medical imaging, and others. But the interest in artificial intelligence applied to biology is not limited to academic research. We are witnessing mega rounds being raised by AI-first drug discovery startups. But let's first clarify, what is artificial intelligence versus machine learning versus deep learning? Artificial intelligence, or AI, is a program that mimics human behavior. It includes robot process automation and artificial general intelligence, which is the hypothetical ability of an agent to understand or learn any intellectual task that a human being can. Machine learning techniques use statistical methods to enable machines to improve performance through the acquisition of experience in terms of data. Here we have methods such as decision trees, support vector machines, neural networks. And deep learning are neural networks with many layers trained on vast amounts of data. They all provide models that learn from examples to give predictions for unseen data. And the tool I would introduce briefly, called Automation Recommendation Tool, that we developed, is one of the machine learning models. So let me introduce the automated recommendation tool. So ART provides predictions with the associated, ART produces a predictive model and recommendations for the next cycle. So the input for ART are a set of proteomics, transcriptomics, or media components, or some other inputs of interest, and the associated response, for example, target molecule production. Given this input, ART produces a predictive model in terms of probabilistic representation, saying, for example, this protein profile has 10% chance of producing 10 milligram per liter. On the other hand, ART uses this model to provide recommendations for the next cycle. So ART provides the predictions with associated uncertainty rather than providing a point estimate. So it characterizes the uncertainty of the provided predictions and recommendations for the next cycle. You can find more information and nitty-gritty details of ART in our publication, and in general, how to use machine learning to guide metabolic engineering in a systematic fashion in the review we published this year. ART core consists of model building and optimization. So given trained data set and the specified objective being maximization, if you would like to improve production of a target molecule, minimization, if you would like to decrease toxicity, for example, or specification, if you're interested in hitting specific levels of your target molecules. So ART first builds the predictive probabilistic model and then proceeds to perform global optimization to provide recommended designs with uncertainty quantification for those recommendations. So we built a predictive model through a novel Bayesian ensemble approach. So ART is an ensemble of several individual scikit-learn models. And the final model is a Bayesian ensemble approach that takes into account predictive accuracy of these individual models and represents the final model in a probabilistic manner. This rigorous quantification of uncertainty enables a principal way to test hypotheses and scenarios in silico and to guide designs of experiments in the next cycle. In the next phase, using this model and sampling-based optimization, ART provides a set of recommendations for the next cycle. And another feature of ART is that it performs optimization of a parameterized function, which accounts for both exploitation and exploration, given in terms of this parameter alpha that balances exploitation and exploration. So for example, this is particularly relevant for early stage cycles, where we recommend setting this parameter to something closer to 1, hence allowing systematic exploration of regions of input space associated to high uncertainty in predictive response. This leads to data acquisition in regions with high uncertainty and improves model accuracy in the subsequent cycles. If the objective is to purely optimize the level of the response variable, for example, during the final cycles, then we suggest setting this parameter close to 0, focusing on exploitation mode. And intermediate results produce combination of both behaviors. So ART is bridging the learn and design phases of the design, build, test, learn cycle. And it can leverage amounts of data in a systematic way. For example, a possible DBTL cycle within Agile Biofoundry or a Joint Bioenergy Institute will be to use DIVA to design and build strains, which then combined with media, we put into fermentations, into our biolector fermentation platform that can take samples automatically. Oh, okay, my cursor is working. We then we then measure the metabolism of the strains through mass spectrometry. Typically, we measure transcripts, proteins, metabolites. We store that data into a repository and use the data with algorithms to learn from them and suggest designs that are predicted to provide better production of biofuels and bioproducts in the next cycle. So we put these designs back into DIVA and do this all over again in a systematic way. How we store data and metadata is critical. Because if there is no data, there is no machine learning. When you're collecting large data sets, Excel sheets are just not practical. For that purpose, we developed the Experiment Data Depot. It is a web-based tool for storing different types of omics data, transcriptomics, proteomics, metabolomics, and metadata. And EDD can be used for different modeling purposes, for data exploration, for interactive visualizations, and data sharing. This is an example of a set of studies in one of our EDD instances, for example, in the Agile Biofoundry. And multi-omics data here can be stored systematically in a centralized repository. And importantly, they can be downloaded easily into a Jupyter Notebook through the REST API. I will now mention some of the examples of using art on real experimental studies. This study concerned the optimization of the complexly regulated aromatic amino acid pathway that produces tryptophan in baker's yeast Saccharomyces cerevisiae. This case highlights the power of combining mechanistic and machine learning models to enhance the predictive power and effectively direct metabolic engineering efforts. The pathway design, in this case, was in terms of selecting which promoters to use for tryptophan production in Saccharomyces cerevisiae with the goal of improving production of tryptophan. So this is the main pathway, all the way from glucose to tryptophan. And we would like to modify promoters in all of the reactions. But since we are limited by the number of promoters that we can use, what we did was we leveraged mechanistic genome scale models to select 5 native genes that will have most impact in the final yield. For those genes, we identified the promoters and created a library by replacing native promoters of these 5 genes. And new CRISPR methods permit one-pot assembly and genomic integration of this library. ART produced predictive model for tryptophan productivity. And as input, we use promoter combination, and for the response, we use biosensor readout that allowed us to phenotypically characterize tryptophan flux in a high throughput fashion. So we used rate of GFP as a proxy for tryptophan productivity to be optimized. ART was trained on these trained data sets and constructed a predictive model for all of the promoter combinations, and then recommended promoter combinations predicted to improve production for the next cycle. As this is biology, things don't always work as expected, and the number of samples to be used by ART was limited by a variety of different errors in the process. So we had 696 well plates for each of the samples. We collected genotype, fluorescence, and growth for 81 time points. This resulted in about 1,700 samples. However, for some of them, they couldn't find genotype information, the PCR didn't work. For some of them, the assemblies failed or samples didn't grow enough. For some, they could not cure the plasmid out, or some of the samples didn't come from the sick population. So all these errors resulted in obtaining about 250 genotypes that forms about 3.2% of all possible genotypes for this particular design space. In the final library, most promoters were well represented in the sample, as we can see here, except of maybe for the third gene, the fourth promoter, the fifth promoter. Also, both good and poor performance were included in the data set. This is important because when you teach somebody, it is important to show them good and bad examples. If we look at the data for productivity, if you look at each gene, so we have the plot for each of the 5 genes here, the production is all over the place. You cannot say much about the correlations and dependencies, except for maybe third gene, where promoter number 4 seems to result in higher production than than for other promoters. But we hope that ART is able to find underlying dependencies of promoter combinations that result in higher production levels. So finally, we train ART, and by looking at the predictive accuracy of the final ensemble model compared to the accuracy of all individual machine learning models, we see that the ensemble model outperforms them at some level. And in terms of mean absolute error or R squared. And then ART suggested new promoter designs by using this trained probabilistic model. These are the set of promoters we recommended. They went back to the lab and built those trains. And finally, the results provided recommendations that improved productivity by 17% over the best one in the library and 106 over the base reference train. Furthermore, ART has been applied to other published studies. ART was used to improve limonene production in E. coli by balancing the pathway in terms of protein expressions. It was used to hit target metabolite levels to achieve hoppy flavor in hopless beer by using also proteomics data obtained from bioengineering Saccharomyces cerevisiae. Another example, which wasn't as a successful story, which we explain in the publication, was bioengineering E. coli to improve the decanol production. But also ART is currently being used to design pathways and media compositions for a variety of organisms and target molecules. In these particular examples, I mentioned proteomics and promoter combinations. The ART can be used for other types of variables that take part in a bioengineering process. So how could one use ART? ART can be used via Jupyter Notebooks as a Python library, or a user-friendly front end at art.agileBiofoundry.org. Both have their pros and cons, such as coding experience required and flexibility in use cases. We provide a free non-commercial use for academic institutions, and more information on licensing and different tutorials can be found on ART's website at art.lbl.gov. I will now switch to another tool we have been developing that concerns learning the kinetics of metabolism. For these purposes, we use deep learning, leveraging from multi-omics data set that we have been generating in collaboration with company Lygos, located in Berkeley, in our neighborhood. So the goal of this project was to improve production of malonic acid through multiple design, build, test, learn cycles. We are generating and leveraging multi-omics data sets to train machine learning models, and then to make predictions on how to engineer Pichia kudrevisi strains to improve malonic acid production. So why malonic acid? Malonic acid has been in use in synthetic chemistry for over 150 years, but it is difficult to produce it from petrochemistry, and production is largely driven by foreign supplies. However, its market share is significant. So kinetic modeling attempts to predict metabolic behavior to produce a desired outcome. So for example, we will have a strain, we would perform experiments in the fermentation platforms, we would measure the metabolism and then collect data. And then we would try to create models of the kinetics and provide recommendations for the next cycle. These models are usually cumbersome Cayley-Smanton equations, but when predictions fail, it becomes complicated to systematically change these equations. So what to do then? Our approach is to use machine learning because it is designed to improve performance as more data is added. And in this case too, we use supervised machine learning, where we have a set of inputs and the associated response, and the predictive model is aimed to predict response for unseen input data. In this particular case, we are using proteomics as input and metabolomics as response. We are performing 6 DBTL cycles in total, and each cycle produces a new set of 24 strains to improve in the next cycle. So we start from initial strains, we perform fermentation in Amber 250 fermentation platform, we collect metabolomics and proteomics, we upload our data into experiment data depot, we train our deep learning models, we provide recommended strains for the new cycle, and proceed to the subsequent design, build, test, learn cycle. And the aim is to perform 6 of these cycles. So to our knowledge, we are producing the largest public multi-omics data set to date. We're producing about 80,000 data points per DBTL cycle. So we are collecting 63 proteins measurements, metabolomics of 72 metabolites. We are collecting 11 HPLC metabolite measurements and 6 measurements from bioreactors from Amber 250. The most important one that we use as our response variable is a malonic acid key performance index. So it's a metric that represents malonic acid production. We also collect pH, dry cell weight, dissolved oxygen, CO2, O2, and so on, OD measurements. And we do this for 24 strains in triplicates, and we collect data for 8 time points. Our machine learning workflow with these multi-omics data consists of the following. So Lygos is building strains and performing fermentations. We then measure metabolomics and proteomics at Agile Biofoundry and Lygos. The raw data is imported into experiment data depot, and then we proceed with exploration and quality check. So this is an iterative process where we are trying to curate the data set until we finally have a final data set that has all the units at place. We take care of missing data. So this is a very cumbersome part of the pipeline. So we invest a lot of care in making sure we have the final curated data set that will then be imported into our machine learning part of the pipeline. So once we have this pre-processed multi-omics data set, we train our machine learning tools using high-performance computing platforms, and then we provide predictions in terms of recommendations, and then we also get some feedback from our collaborators on the nature of these recommendations. So how likely they are to be produced, how difficult are those genes, something they would expect or not, and so on. So as I mentioned, data quality, checking and pre-processing is critical for downstream analysis, because we need high quality data with relatively low variations to ensure the confidence in our predictions as well as recommendations. Further on, model fitting requires thorough data preparation. So once we have the curated data set, we still need to prepare and check data sets for the machine learning part of the pipeline. So what we do, we first interpolate these 8 time points to 63 hourly time points. We have to define what are training, what are test strains. We have to define what are inputs and response. In our pipeline, we allow some flexibility for future, so we can change this. We can investigate different proteomics, different metabolomics, or even different types of data. We also have to take care of the data standardization, and for the purpose of validation of our model accuracy, we have to take care of the proper train and validation partitioning. So we predict response time series of a malonic acid key performance index, KPI, from input time series of proteins. So we have 63 time points, 63 proteins, and 63 instances. We build 3 types of deep neural networks, which are dense neural networks, recurrent neural networks, gated recurrent unit, RNNs. So these 3 types of methods form our final ensemble model that is used to provide predictions. But we also use a partial least square model to prioritize proteins for producing recommendations in the next phase. So by doing this, we allow that some of the artificial neural network layers account for core metabolism, while other specific layers focus on particular metabolic products. So if we look at the performance of our final ensemble model, in this graph, we see for trained strains in gray and test strains in pinkish, we see time series of predictions in red and observations in green. So by looking at these dynamics, we are dynamics of the malonic acid KPI as a function of time, we see that the ensemble model is performing acceptably, very well for the trained strains, of course, and reasonably well for the test strains. If we look a little bit more and quantify this accuracy, we see that the ensemble model shows very good agreement between predictions versus observations. So here, having points on the diagonal line is ideal, meaning that the observed malonic acid KPI coincides with the predicted malonic acid KPI. We are very pleased to see these results on this particularly small data set after the first cycle. Nevertheless, if we look at the predictive accuracy for only the last time point for the malonic acid KPI, we see that the ensemble model accurately predicts this point. So the test strains are represented in red and train in black. We observe very high R-square and low mean absolute error. Once we have this model, we proceed to generate recommendations. And in this case, we generate recommendations by exploring allowable modifications in protein space. So allowable meaning what kind of modifications Lygos is able to build. In this particular case, possible modifications of protein expression are up to 3 increase expressions. We call this up to 1 in combination with maximum of 3 increase expression. We can include 1 knockout, or we can include 1 knockdown, which is a reduced expression of 1 protein in combination with the maximum of 2 increased expressions. So we try to explore these kind of modifications for each of the strains, and this translates to 10 possible types of modification. So either single down or knockout or increased expression, or combinations of those with increased expression. So we used partial least squares to guide the exploration of the possible modifications and then to make recommendations. So we build the partial least square model, here plotted, and then we use this model to create an ordered list of proteins that we would consider for these types of modifications. So by looking at the weights in this model and the list, we are considering proteins from the beginning of the list for overexpression, because this particular metric is positively associated to the increased response variable, malonic acid KPI. And inversely, we use proteins from the back of the list for knockouts and knockdowns. So we perform this computation in parallel. This takes about 10 hours of computation in parallel. And finally, we provide recommendations, multiple recommendations for each of the strains in terms of these modifications. So we provide which protein and which modification, and we provide a factor of increase response variable in this case. So we can provide, we can design our model to predict and to order the recommendations in terms of factor of improvement of the base strain, or just a simple absolute value of the product concentration. This corresponds to the approach of doing more exploration in the earlier cycle. So if we purely want to increase to put every strain to the next level and increase the production, rather than just focusing on any strain and simply increasing the production of the molecule, regardless of the initial design. We are now at the middle of the DBTL2, and fingers crossed, we are waiting for the experimental results on the recommendations. So we still don't know how well our recommendations perform. We only know, we only have the result that I showed you about the predictive accuracy, but we still don't have experimental results on the designs that we provided. And we hope to have them very soon, in the next few weeks or a month. And finally, yes, I would like to end with the idea that synthetic biology needs predictive power to enhance its global impact. And we have created machine learning tools that leverage increasingly available amounts of data to predict responses, for example, production, and suggest the next steps. We created automated recommendation tool that was successfully used to design pathway for tryptophan production, but is also being used to design pathway and media compositions for a variety of organisms and target molecules. We also created an alternative to traditional kinetic modeling, kinetic learning, which is purely data-driven approach, not requiring deep knowledge of the pathway and final product, but providing a general method applicable to any host, pathway, or metabolite. We develop pipelines for data processing, training multiple neural networks able to predict product dynamics, and generating actionable recommendations that are predictive to improve malonic acid production. And for this, we are producing a very large data set of about 480,000 data points to train these models. And so far, predictions using these models look good, and we are waiting for the testing of the proposed recommendations. We also hope that multi-omics data collection cost is likely to drop in time, and that these approaches may become commonplace. Many people have contributed to the development of these tools, and I would like to acknowledge their efforts. First of all, our quantitative metabolic modeling group led by Hector Garcia Martin, and many members of the group who worked on development of these tools. Also, we have very close collaboration with Bioinformatics group led by Nathan Hilson. We collaborate closely with proteomics and metabolomics facilities to obtain data with DIVA and automation team. We have a great IT support, and I would also like to acknowledge our collaborators from DTU in Denmark, in particular, and LIGOS, as well as our other collaborators at Joint Bioenergy Institute and Agile Biofoundry. And I would like to thank you all for your attention, for being here, and I'm happy to take any questions you might have now.
And thank you so much, Dr. Tijana. And now we're going to move on to the questions. We are having now an open discussion. So, Prashant, you have the word, I think.
Yeah, absolutely. Yeah, thank you so much, Tijana. Really appreciate it. This is a great talk. And it's really great to see some how art is progressing and how it's having a lot of impact as well. So that's absolutely great. So actually, yeah, I think it'd be great to have an open discussion session here. So I know there's a question here already. Michael, if you want to ask this question, and we can just get started, or I'm also happy to read it out.
Sounds great.
Sure, I can just ask. Thanks very much, Tijana, for the talk. That was really great and insightful. Could you comment on how you expect your method to improve, let's say, production of a product over time? So what I mean is, do you see a trade-off, or do you expect a trade-off between the number of cycles of redesign compared to how much of a product you would produce? And maybe you can comment whether you expect to see that incrementally, or whether you expect that to be a large step change, like one round and you get a large increase in production of a product.
Great question. Thank you for this question. So there are several things to consider when designing an experiment, and we are really trying to advocate for close collaboration between computational people and experimental scientists, because this is exactly this collaboration that's helping us to get to those answers. So realizing from the beginning, what is the objective, what are the resources, very importantly, and yes, further on, what are the timelines, expected deadlines, and so on. Very importantly, we first have to define what is the question, what are we trying to optimize, and what are the actionable items that we can, what are the designs that we can act upon, and how many of those. So these first questions set the ground for defining how many instances we are able to create, and what would actually be then, how we could kind of estimate how our models would perform. For example, if you have interest in hundreds of proteins that you believe would affect the production, you really need large amounts of data. Let's say, in my opinion, you would need at least 1,000 data points. But then we can still try to work with less. In that case, we try to start the initial round and then iteratively obtain these data sets, and then iteratively our model will improve. So all this depends on the resources. Of course, more data you have, more accurate the model would be, and the recommendations would be more accurate. The same thing applies. If you have more ambitious question on how many variables you would like to explore, you would need to provide larger data sets. Of course, in the initial designs, the model knows nothing about the space. So we first try to explore random draws by using Latin hypercube, for example, meaning that there is no learning yet. But then from the first cycle on, where we do have labeled data sets, meaning we have both input and response, we start learning, and the model is expected to incrementally improve the production. This, of course, this depends if we are talking about art, it depends on the mode you are performing. So if you are doing exploration or pure exploitation and so on. So there are different considerations to take into account when designing an experiment.
Thanks very much.
Thanks, Tijana. Actually, I kind of have a question that's sort of related to that. I mean, this is obviously very interesting. What I'm kind of curious about is he had mentioned how art is been used in multiple different examples or case studies. So what I'm kind of curious is, along these lines, how easy or difficult is it to actually change like the underlying, let's say, the model or the objective function that you're trying to train? Obviously, the data is important and you need that to train your model. But then I'm kind of curious, especially when you're looking at the Bayesian optimization side of it, like for instance, how easy is it to change the objective function, which in turn would, affect your exploration or exploitation phase, for instance?
Mm-hmm.
Kind of curious about that.
Yeah, great question. The beauty of art, well, its blessing and a curse, is that it is a general tool. So there is no need for you to change anything. Art is agnostic when it comes to the host, to the target molecule. Objective is something that you just specify a type of an objective, either maximization, minimization, or specification, nothing more. You provide the parameters in terms of a string saying maximization. That's all that it happens. So for you to use art on a completely different problem with a new bug, you do not need to change anything, but you need to provide sufficiently large training data set, because that's only thing that art needs to provide the predictive model and recommendations.
All right, sounds great, thanks. I think we have Eric next and then Giovanni after that. Eric?
Hey, thanks. Really, really great talk. I have a question about the second half. We were discussing the model generation. You have this proteomic data and the output's going to be these metabolomic profiles. In this collaboration with LIGOS, how difficult is it where, say you do this first round and now you have these recommendations, and the recommendation is this: please produce this proteomic landscape for me, because I think this is going to lead to the best metabolite. There's almost the question of, great, well, how do I produce that proteomic profile? What genes do I have to manipulate? How have you navigated those obstacles in it? Are those severe dead ends, or how are they trying to approach that? What does that look like?
Excellent question. This applies to other studies that I also mentioned that all work in terms of protein expressions. So yes, the question is, what kind of designs can you provide such that they're actionable? And you can only achieve as much in achieving those specific targets. So here is where the iterative approach comes handy, because we're not claiming art is almighty and it can provide accurate predictions, especially because those even levels are very difficult to design, as opposed to promoters, for example, where you really know which promoters you're going to use. That was an excellent point. So in this particular collaboration, of course, there were challenges in building those. We are now ending the build phase of those profiles, but I know so this is done on the side of LIGOS. I know there have been struggles. For example, they could not express a particular promoter, so they would identify genes and then target them, build them with certain promoters. But so there were problems with some particular genes more than with others. So they're trying to find ways to go around iteratively, and yes, hoping that eventually results definitely improve by every trial. But definitely that's a challenge.
Cool, thank you.
And we're happy that they were able to build all of those eventually, and they even plan to build in advance different combinations such that they can be ready for the next recommendations.
Great, thanks.
Thank you. We have Giovanni and then Alejandro.
I just want to know if we can use design of experiments together with machine learning for design, build, test, and learn?
We anyway use it. We use design of experiments for the initial cycle, where there is still no knowledge on the system. So it is integrated with art. It's exactly the same interface. You will just specify that this is your initial cycle. And then in the next phases, you will proceed with learning, with different types of machine learning models that are training, that are training their parameters to provide a final model by using the trained data set.
Thank you. Alejandro?
Yes, thanks, Diana, for the presentation. It's nice to see people already doing almost like the complete DBTL cycle, so very nice.
Yes, and multiple.
I was wondering the recommendation, at what level is the recommendation? Because related to the previous question, are you telling the people which is building, you need to build exactly this? So are you giving the DNA sequence, or what is the level of recommendation that you are giving in terms of that, and also in terms of the experimental part? What kind of experiments and measurements, and when do you need to take the measurement and stuff like that?
Yeah, good question. So depending on a study, so for example, in some of the studies, we would provide recommendations for the protein expression levels case. We would just say, okay, these are the numbers of expressions predicted to improve. However, as we just discussed, this is very difficult to achieve. For the Lygos case, we provide these recommendations in terms of overexpression. So we assume that expression is doubled. We then for other proteins, we say you need to knock down the expression, assuming that it's half of the expression, or knock out, so remove that gene. So these are the three types of modifications that we provide, because this was agreed at the very beginning when we all sat down, brainstorm how we are going to design these and what are the capabilities. So it's given in those terms. And yes, in terms of time points, our model assumes that these modifications are done at the very beginning. So so this would be the expression level before fermentation starts. And then the model assumes that these expressions propagate in time. I know this is a very strong assumption, but for now we work in these kind of constraints.
Okay, thank you. And one small one, if I can. Are you thinking on trying to fill this gap between making the recommendation at the level of expression that you need, or assuming that you will be having a double level, to going to the DNA sequence level recommendation?
We are definitely interested in looking at that. However, for that, you would need the input design space to be defined in terms of DNA combinations. So recommendations will be provided in exactly the same form as the input data set. In this case, we had protein expression, so we could not work with anything else than protein expressions. If we work with sequencing and have the associated phenotype, yes, we could then build the model. However, of course, sequences are much more highly dimensional spaces, and meaning that for those, we need much larger data sets to be able to train our models.
Thank you.
Leon?
Hi, Tijana. Thank you for this great talk. I wonder, because art contains a lot of different models, which is like compiled into these ensemble modeling techniques. So I guess there are all black boxes, and I wonder if in art there are some automatic extraction of the key features where you can like explore these black boxes to get some mechanistic insights on what's actually happening during the experiment, or if it's still something to do outside of art.
Excellent question. So far, no, we haven't looked at that, but we definitely are thinking about combining art with other approaches to extract some knowledge. So ultimately, the best way to advance the knowledge would be to combine existing knowledge and data generated. So art is for the moment is purely data driven. We can look at the different weights of these individual models, but this is all done outside if the researcher is interested in really looking the inners, but is not something that is provided to the user.
Okay,
Great question.
All right, thank you. If anyone else has any questions, please let me know. But in the meantime, I actually have one quick question, actually. So it looks like art does a lot of its, I mean, a lot of the data that comes into art is via proteomics. So one thing that I'm kind of curious about this is how does art deal with, you know, things like missingness of data? Like, at least in my experience, one, if not many, one of the biggest challenges in proteomics is you often have, like, there's a lot of missing reads, for instance, like missing protein information. So is there a way to like deal with that, or has that not been an issue so far? I'm just kind of curious.
Yes, as surprising as it sounds, we did not have those issues for proteomics. We are very proud to have great proteomics facilities at our institutions. So art didn't have those issues so far. When it comes to kinetic learning side, where we had time series proteomics, we did have some missing data, but since we performed interpolation of those time series, we were able to go around some of those missing points.
All right, thanks. Franco?
Yeah, thanks, Tijana, for a great talk. I was wondering, now that you were talking about the kinetic models and data-driven models that you use, is there a way to include that kinetic data that you might know about the system to help with the predictions?
You refer to the second part?
Yeah, to the last part
into
of the
art.
talk.
So, well, the second part also assumes that we have no knowledge about the kinetics. So this approach is purely data-driven. So instead of incorporating the known equations or something known about the system, we are letting the model learn purely from data, and then constructing those dynamics, those time curves. Yes, as the question earlier, definitely our goal is to, in future development, methods that would incorporate both mechanistic knowledge with generated data to improve our knowledge on the system.
Great, thank you.
Thank you. And I think we have one more question from Giovanni.
How do we know which technique should we use when we deal with things like bias, variance, or overfitting? Like, when do we use things like partial least squares, or when do we try something else?
So I guess you're asking about problem in general. So how to deal with overfitting, you always have to take care of the train and test and validation partition, so that you make sure that you are not, your model would be able to generalize. So by doing this partitioning, you really have to take good care by making sure that all replicates, for example, belong to only one partition, because you might get some overfitting if some of your replicates belong in the trained data set and others belong in the test. That means that your model already seen those data and it's not a fair comparison to you. So you might have some overestimated predictive accuracy. So for example, we do cross-validation when we report our tenfold cross-validation, when we report our performance accuracy. And yes, I think that's the main thing, how you would deal with that. I don't know if there was something else in the question.
I guess, yeah, that answered it. So yeah, I think we probably have maybe time for maybe 1 or 2 more questions. So we'll just wait for a few seconds to see if anyone has any questions there. Well, all right, if not, well, I just want to thank Tijana again for such a great talk. Thank you so much. It was lovely to have you as our keynote speaker, and it's really great to see, you know, the work that you've been pioneering about how machine learning is really making a great impact in synthetic biology. So thank you so much. Really appreciate it.
Thank you so much, everybody, for being here and for your great questions. I hope you all will enjoy the rest of the conference. Thank you all.